Assalamu alaikum class. Today I am going to give you a very basic and brief description of the special sense of taste. Uh, Sensory system is a separate entity of the nervous system which makes sense of your senses. And there are generally five senses, which are, as you can see here, these are touch, sight, hearing, smell, and taste. So, what are the special senses? You see, the senses that have specialized organs devoted to them are called special senses. In contrast, the general senses, or you call them the somatic sense, it does not has a specialized organ which is specially there for a single modality of the sensation. Now, special senses, they are vision, hearing and balance, smell and taste. While the example of a somatic sense is touch, which is coming from all over the body. Now you see that for this touch sensation, the types of receptors which are present in our skin, they are mechanoreceptors, and they are thermoreceptors and the nociceptors. The nociceptors, they are for detecting painful stimuli, while thermoreceptors, as you can understand, is for temperature detection, any change in the temperature. While the mechanical receptors, these mechanoreceptors, they are there uh, for touch, no matter is it fine or crude touch. Then they also sense pressure and vibration. There are many shapes of these skin receptors. They could be onion shaped. There are Pacinian corpuscles, Meissner corpuscles, and we can say that the nerve endings for these somatic senses, they are specialized, but they can be used for the detection of touch, temperature, and pain sensation. So, a single organ and a single receptor shape is not dedicated to one single modality of the sensory part. If we move on to the special senses, you see, for sight, we have got eyes, and they have got photoreceptors which are given a special name. There are rods and then there are cones which are present in the retina of eye. Then, for the sense of hearing, and not only hearing but also for the sense of balance and equilibrium, we have got mechanical receptors in our ears which are called stereocilia. For taste and smell, the type of the receptor is the chemical type. So, for the sense of smell, we have got olfactory cells in our nose, and for the taste, there are gustatory cells present on the tongue.
Animals, including humans, depend on the chemical senses to help identify nourishment, like the sweetness of honey or the aroma of pizza. Then the chemical senses, they also help identify nauseous substances such as the bitterness of plant alkaloids, or they also help identify the suitability of a mate. Chemical sensation is the oldest and most common of the sensory systems. Even brainless bacteria can detect and tumble towards a favorable food source. Humans live in a sea of air full of volatile chemicals. We put chemicals into our mouths for a variety of reasons, and we carry a complex sea within us in the form of blood and other fluids that bathe our cells. We have specialized detection systems for the chemicals in each environment. The mechanisms of chemical sensation that originally evolved to detect environmental substances now form the basis for chemical communication between cells and organs using hormones and neurotransmitters. Virtually every cell in every organism is responsive to many chemicals. Many types of chemically sensitive cells, means the chemoreceptors, they are distributed throughout our body. For example, some nerve endings in skin and mucous membranes, they warn us of irritating chemicals. A wide range of chemoreceptors report subconsciously and consciously about our internal state. Nerve endings in the digestive organs detect many types of ingested substances. Receptors in arteries of the neck measure carbon dioxide and oxygen levels in our blood. And sensory endings in muscles, they respond to acidity, giving us the burning feeling that comes with the exertion and oxygen debt. Gustation and olfaction have a similar task, means the detection of environmental chemicals. In fact, only by using both senses can the nervous system perceive flavor. Gustation and olfaction have unusually strong and direct connections with our most basic internal needs, including thirst, hunger, emotion, sex, and certain forms of memory. However, the systems of gustation and olfaction are separate and different regarding the structures and mechanisms of their chemoreceptors, the gross organization.
Today we will start with gustation, which is the sensation of taste. Since Physiology is all about function. So we will focus on the main functions of the sensation of taste. Taste is a relative group that serves primarily as a gatekeeper to the gastrointestinal tract. In addition, it is used to separate undesirable foods from the pleasant ones. Another important function of gustation is to avoid lethal foods. Now, the question is that how do we acquire the taste fully? The answer is that it is mainly a function of taste buds. Factually speaking, taste is a cumulative sense, but one's sense of smell also contributes strongly to the taste perception. Then the texture of food is detected by tactual senses of mouth and presence of substances in food that stimulate the pain endings, such as pepper, greatly alter the taste experience. Now, I'm going to tell you the role of taste buds, which are ovoid bodies with a diameter of 50 to 70 microns. In adults, about 3,000 to 10,000 taste buds are present, while the number is more in children. But in old age, many taste buds they degenerate and the sensitivity of taste becomes weak. And now exploring other species, we have come to know that the insects they have taste organs in their feet, and in their antenna, and even in their mouth parts. A taste bud, which is a functional unit of taste sensation, is composed of the receptor cells and the sustentacular cells. These receptor cells they are modified epithelial cells, and they are about 50 in number. There are only few sustentacular cells which are supporting in nature, and then many sensory nerve fibers they are intertwined among the cell bodies. Now let's have a look at the physiologic structure of taste bud. Taste bud is basically a bundle of taste receptor cells whose supporting cells are embedded in the covering of papillae. Categorically, cells of a taste bud they are divided into four groups: type 1, 2, 3, and type 4. Now, type 1 and type 4 they are the supporting basal cells, while type 1, 2, and 3 they have projections which are called microvilli. These microvilli they project into an opening in the epithelium covering the tongue, while the neck of each cell is attached to the neck of others. It is noteworthy that there are tight junctions between epithelial cells and the neck portion of type 1, 2, and 3 cells, so that only the tip of these cells they are exposed to any fluid in the oral cavity. Then the cells of taste buds they undergo constant cycle of growth, apoptosis, and regeneration. But why this regeneration is required? Reason is most receptors are carefully sheltered from the direct exposure to the environment, but the taste receptor cells, by virtue of their task, frequently they come into contact with the potent chemicals, so they have to be replaced continuously. Now, during this process, the epithelial cells surrounding the taste bud, they differentiate first into the supporting cells and then into the receptor cells. I already mentioned about the taste pore, which is formed by the apical surfaces of taste cells. And taste hair protrude from this pore, and that's how surface for tasted molecules is provided by a taste hair or a microvillus. Now, taste bud, which are smaller and closer to the tip of the tongue and larger toward the back, they are found in relation to tongue papillae. There are four types of papillae: fungiform, circumvallate, foliate, and filiform. Filiform papillae are small and conical shaped papillae. The interesting thing is that they are situated over the dorsum of the tongue, but they have no taste buds. Fungiform papillae are located on the anterior two-third of tongue, and these are round in shape, and the number of taste buds in each is moderate, which is up to 10. Then come circumvallate papillae, which form a V shape on the posterior one third of tongue. These are large structures, and each papilla contains many taste buds up to 100. As far as foliate papillae are concerned, they are located along lateral margins of tongue. Now, there are some other locations also for the presence of a few taste buds, and these are palate, pharynx, tonsils, epiglottis, and proximal esophagus. Now, there are five primary taste sensations, which are sour, salty, sweet, umami, bitter. And now we will discuss each one by one. Sour taste is caused by acidic substances, for which the receptor involved is called epithelial sodium channel. Although the proton which enters the receptor, that is hydrogen ion. There is another channel involved, and that is nucleotide gated cation channel. Now, the intensity of sour taste is approximately proportional to the logarithm of hydrogen ion concentration, which means the more acidic the food is, the stronger the sour sensation will be. For salty taste, the receptor involved is again epithelial sodium channel, and the cations involved are of ionized salts, mainly by sodium ion concentration. Then the anions, they also contribute to a lesser extent. Quality of salty taste varies from one salt to another, because some salts they elicit other taste sensations also in addition to the saltiness. Sweet taste is not only caused by a single class of chemicals. Now, some of the type of chemicals that cause this taste include sugars, glycols, alcohols, aldehydes, ketones, amides, esters, some amino acids, and some small proteins. Then there are sulfonic acids and the halogenated acids. Uh, you may have noticed that most of the substances that cause a sweet taste, they are organic chemicals, while the inorganic substances which produce sweet taste are lead and beryllium. Now here is the twist. Like slight changes in chemical structure, such as addition of a simple radical, it can often change the substance from sweet to bitter. Hmm. Umami taste designates a pleasant taste sensation. Umami is a Japanese word meaning delicious. It is a qualitatively different sensation from sour, salty, sweet, or bitter, and it serves as a marker for a desirable and nutritionally protein rich food. This taste is triggered by the presence of amino acids, especially L-glutamate, for example, meat extract or aging cheese. Receptor for this taste is metabotropic, whose activation is intensified by guanosine and inosine monophosphate. Now, bitter taste is also not caused by only a single type of chemical agent. And substances that give bitter taste are almost entirely organic substances. There are two particular classes of substances which cause bitter taste, and these are alkaloids and long chain nitrogen containing items. Examples of these substances are quinine, caffeine, strychnine, nicotine. Interestingly, there are many plants, fungi, and some animals which produce bitter toxins as a natural defense mechanism. As far as the detectors are the topic of interest, you must know that most bitter tastants they are detected by G protein coupled receptors. Now, taste cells that detect bitter possess around 50 to 100 bitter receptors. Each of these receptors they respond to a different flavor of bitter. Since each cell has a diverse family of receptors, a wide variety of unrelated chemicals all could taste bitter despite their diverse structures. And hence, this mechanism it expands the ability of a receptor to detect a wide range of potentially harmful chemicals. Okay, now now I'm going to give you important examples of some common bitter substances. One of them is quinine, which originally is bitter tasting toxin with anti-malarial properties, and it is extracted from a tree bark. For eliciting the taste, it can block most classes of potassium channels and cause non-specific membrane depolarization. Then there are some substances which uh, initially taste sweet but have a bitter aftertaste. For example, there is a saccharine, but this characteristic makes the substance objectionable to some people. You must have experienced that there are other taste like sensations. For example, taste of fat constitute a sixth basic taste, but its transduction mechanisms they are not fully delineated. Now, can you think of any other taste like sensation which actually are not taste? Well, there are these chemical sensations that mimic hot or cold. For example, the burning sensation associated with chili pepper. And then there is menthol. Now, these are not tastes, but since they are mediated by the somatosensory pathways located in the oral cavity and nasal passage, they are considered taste sensations. Just to regain your attention, I will include another taste like sensation, and it is the fizzy sensation on your tongue from carbonated drinks. This is not caused by the tiny bubbles popping. The fizz, this is the actual taste of carbon dioxide. The type of receptor protein and its specific action in each taste villus determines the type of taste that will be perceived. Now, I give you a few examples. For sodium and hydrogen ions, receptors open specific ion channels in apical membranes of taste cells. And for sweet and bitter taste, portion of receptor protein that protrude through apical membrane activates second messenger transmitter substances, which result in intercellular chemical changes, eliciting the taste signal. Uh, well, I feel that you still have a question that how the difference in the taste is appreciated. You see, each taste bud typically responds to only one of the five primary taste substances, except when an item is present in very high concentration. This discrimination is coded by patterns of activity in various taste bud receptors. So we can say that each receptor cell responds in varying degrees to all the primary tastes, but in generally preferentially responsive to one of the taste modalities. That's how the discrimination depends on subtle differences in the stimulation patterns of all taste buds. And here are 13 taste receptors. Now we are going to move on to the taste transduction, which is a process in which taste chemoreceptors convert the chemical energy into action potentials in taste nerve fiber. If I put it in few words, what happens that dissolved substances act on exposed microvilli, which are also called taste hair or cilia, and this leads to the development of receptor potential and ultimately to the generation of action potential. Now, how the receptor potential is initiated? Like most sensory receptor cells, membrane of taste cell is negatively charged on inside, and application of taste substances cause partial loss of the negative potential. Now, this decrease in potential is approximately proportional to the logarithm of the concentration of stimulating substance, while the change in the electric potential That is called receptor potential. Now let's revise how the receptor potential is generated. You see, there is application of a substance to be tasted, which results in a depolarization of the receptor cell by opening ion-specific channels, and there is a response in the associated nerve fibers. These are just thresholds for different tastes. Now we will have a look at the mechanism of receptor stimulation for different taste sensations. In case of sour, what happens is that there is a presence of free hydrogen ions in acid. This hydrogen ion blocks the potassium ion channel and results in decrease in the passive movement of potassium out of the cell. And that's how a reduction in internal negativity occurs. While presence of salt leads to the entry of positively charged sodium ion through specialized channels and resulting in receptor depolarization. In case of sweet taste, presence of glucose leads to the activation of cyclic AMP second messenger pathway, which results in phosphorylation and a blockage of the potassium channels. Now the bitter tastent, it causes the activation of G protein and phospholipase C messenger system and results in calcium release. Now let's have a look at how the taste nerve is excited. You must know that taste nerve fibers, they form a branching terminal network. This network is interwoven around the bodies of taste cells, while some of these fibers they invaginate into the folds of taste cell membrane. Now, in the next step, many neurotransmitter vesicles they form beneath the cell membrane near the fibers, and the release of the neurotransmitter substance excites the nerve fiber endings. Now, here we mustn't forget about the adaptation of taste sensation, which is achieved by a strong immediate signal in the taste nerve and a weaker continuous signal later on. It means that on first application of the taste stimulus, the rate of discharge of nerve fibers rises to a peak in a small fraction of a second. But then adaptation occurs within a few seconds, as a result of which the rate of discharge of impulses falls back to a lower steady level. But keep in mind, this taste adaptation occurs both at receptors and at the CNS level. Well, let's start with the transmission of the signals into CNS. First order neurons of taste pathway, they are in the nuclei of three different cranial nerves, while the dendrites of these neurons, they are distributed to the taste buds. So from the anterior two-third of the tongue, these signals they travel in the branches of trigeminal nerve, and then they join the chorda tympani, which is the branch of the facial nerve. And from the posterior one-third of tongue, the signals they are carried by the fibers in the glossopharyngeal nerve, From epiglottis and from other areas, the signals they are carried within the branches of vagus nerve. Now, these efferent signals through the axons of first order neurons enter into the nucleus of Tractus solitarius, which is located in the brainstem, precisely in the medulla oblongata, through the solitary tract. It means that the neurons of Tractus solitarius, they are second order neuron. Axons of these, they run through the medial lemniscus. Next, the third order neurons, they are in the postoventral nucleus of thalamus. So the axons, they pass rostrally to the ventromedial nucleus of thalamus. And then the axons from the third order neuron, they project into the uh, parietal lobe. And that's how the signals, they finally reach the cerebral cortex. Final taste perception is achieved in the ventral region of postcentral gyrus, which curls into lateral fissure of the cerebral cortex. In other words, you can say that the taste center that is located in the opercular insular cortex, which means in lower part of the postcentral gyrus, which receives cutaneous sensations from the face also. Now, there is a unique feature of our gustatory pathway that is unlike most sensory input. Gustatory pathways, they are primarily uncrossed, and that will mean the taste fibers, they do not have an independent cortical projection. Pathway for salivation, which is a taste reflex, it is relatively simple because the fibers here, they course from the solitary tract directly to the superior and inferior salivatory nuclei, and activation of saliva secretion, that is mediated by preganglionic parasympathetic fibers to superior and inferior salivatory nuclei, and then postganglionic fibers to submandibular, sublingual, and parotid glands. On average, 30 ounces of saliva is secreted every 24 hours, but any form of sympathetic activation, it can cause dry mouth. Taste preference simply means that an animal will choose certain type of foods over others, and the preference, it often change in accord with the body's need for certain specific substances. Now note that the phenomenon of taste preference or taste aversion, it results from a mechanism located in central nervous system, not in the taste receptors. These are the abnormalities of taste sensation, and we will discuss each of them one by one. Adrusia is a total loss of taste. It may be permanent or temporary. Lesion in a facial nerve, chorda tympani, or mandibular division of trigeminal nerve causes the loss of taste in anterior two-third of tongue, while lesion in a glossopharyngeal nerve leads to loss of taste in the posterior one-third of tongue. Temporary ageusia occurs due to certain drugs such as captopril, penicillamine, or the substances containing sulfhydryl group. Ageusia is uncommon except in patients with the Sjogren syndrome. These patients, they suffer from an autoimmune disease that impairs the exocrine gland function, including the salivary glands. And you all know that saliva is required to carry the taste buds in dissolved form through the taste bud pore. Next is a hypodusia, which is a decrease in the taste sensation, and it is due to increase in the threshold for different taste sensations. Then there is metallic dysgeusia, uh, which is actually a persistent metallic taste, and it is considered a common side effect of antifungals and many antibiotics such as tetracyclines and metronidazole. Then comes the taste blindness, which is a rare genetic disorder where there is inability to recognize the substances by taste. Finally, there is dysgeusia, which is a disturbance in taste sensation in which paroxysmal, unpleasant hallucinations of taste and smell occur. Condition in which dysgeusia present is the temporal lobe syndrome, especially when interior region of temporal lobe is affected. So that was all regarding gustation, and that's the end of special senses physiology as well. So good luck memorizing it.